Hello, we are Team 23089, understanding the normal aging brain so that the puzzle of Alzheimer's can be solved. We are mentored and sponsored by Don McDonald, and our project will help aid researchers at the Evelyn F. McKnight Brain Institute. My name is Claire Peterson. I'm a biosystems engineer and the team lead.
I'm Alec Mitchell. I'm an optical engineer and procurement lead.
I'm Elizabeth Conacher, and I'm the computer engineering lead.
I'm Jerry Flores, and I'm the mechanical engineer.
I'm Gabriel Marquez, and I'm the systems engineer.
And I'm Efrain Torres, and I'm a computer engineer.
Understanding the normal aging brain is necessary before beginning to understand the brain of someone with Alzheimer's disease. Researchers at the Evelyn F. McKnight Brain Institute are studying place cells and grid cells to further understand navigational irregularities and difficulties in the brain as it ages. This video shows a rat navigating around the box to retrieve food that is being dropped in. Shown here is the firing of a single place cell. Each red dot is where that specific cell fires. As you can see in this heat map, the cell overwhelmingly fired when the rat entered this spot. Grid cells fire over multiple points in a given environment, creating a grid-like map. This video depicts the same rat navigating the same space under the same conditions as the previous video. Unlike the last video, there is more than one prominent spot shown through the heat map. The grid cell firing pattern reflects the general configuration of the environment. Together, place cells and grid cells indicate where you are in space, or what could be called your cognitive map.
The Instantaneous Q-Rotation Arena, or ICR Arena, is a novel testing apparatus used to research locational memory in young and old rats. The ICR Arena in its original form can be used to collect place cell data from both young and old rats. It is a cylindrical testing apparatus with visual locational cues projected onto the outside of the chamber. Inside the arena, there is a circular track that is traversed by the rat, which has been trained to stop at specific locations based on the visual cues on the walls. While it navigates, its position is recorded by an infrared camera embedded in the ceiling of the chamber. Throughout the duration of the test, hippocampal place cell data is recorded from the rat via a brain-implanted tetrode. Because those at the Brain Institute are investigating how place cells and grid cells interface with each other and how that intercommunication breaks down with aging, they will greatly benefit from the ICR Arena being able to collect data from both kinds of cells. As this was not originally the case, our team has implemented new subsystems to the ICR Arena that allow researchers to collect a more complete data set from the rats they are testing. We designed or improved upon three main components of the ICR Arena: the open platform, the uniform food distributor, and the food reward robot.
With the addition of grid cell testing for the ICR Arena, a surface for the collection of grid cell data was required to perform such tests. With the arena having an available circular footprint of 1.31 meters in diameter, we decided to use this space for the open platform. Fabricated by hand utilizing workshop tools, the open platform is an anti-static flat circular surface with a near-perfect fit into the available space within the ICR Arena. The platform is made of sinterplastic with a thickness of 9 mm and a diameter of 1.31 meters. It is used for grid cell testing by providing a flat, black, uniform navigation surface that gives no visual cues to the test rat.
In order to meet all requirements baselined in our documentation, the platform was tested and verified to be able to be integrated in a stable manner into the existing system, have a weight manageable by 1 to 2 people, and have the capability of providing a surface for a rat to safely walk on without concerns. Another test conducted in conjunction with the feeder was the ability to scatter dropped sprinkles into a random distribution, which is necessary for testing.
In order to perform grid cell testing, a second subsystem was required, one that would allow for the dispensing of food rewards onto the newly developed open platform. The Uniform Food Distributor is a 3D printed subsystem consisting of 2 individual reward droppers. These are made of food-safe PLA and operate through the rotation of a DC motor linked to the dispensing shaft. Each reward dropper is attached symmetrically across from each other on opposite ends of the ICR Arena's frame. They lie 76 centimeters above from the open platform, where they are loaded with chocolate sprinkles that serve as a reward for the test rat.
The electrical design for the Uniform Food Distributor consists of an Arduino Leonardo, which interfaces with a motor controller board that connects to both droppers, each with a motor and a sensor. The electronic components are contained in an accessible plastic box attached above the ICR Arena with a wire connection to the main computer, allowing the MATLAB GUI to control the droppers. The C++ Arduino software will take serial signals from the GUI to start the test, change the timing, spin either motor, or stop the test.
The Uniform Food Distributor has been tested to demonstrate a full capability of dispersing sprinkles with random distribution across the entire platform. This test will be conducted by utilizing the GUI that controls the software interfacing with the hardware to conduct the dropping of sprinkles. The coverage and random distribution will be verified on the open platform.
While place cell testing is being performed, a food reward robot is secured onto the circular track behind the test subject. The food reward robot keeps pace with the rat during place cell testing and dispenses a liquid food reward if the rat arrives in the correct location. The amount of liquid food reward is based on the accuracy of the rat's stopping location with respect to the specified visual cue projected on the screen. It is important for the robot to make an accurate stop in these tests in order to not only dispense the correct amount of reward, but also maintain a safe distance from the rat so it does not collide with them and startle them. The robot also cleans the circular track with ethanol and collecting rat droppings. We reduced the weight of the robot from 6.25 kilograms to. Through this, we removed unnecessary components, and we gave it a thorough cleaning. These changes were made to allow for better stopping time, improving accuracy for the amount of food reward given to the test subject, and preventing the robot from colliding with them.
The code used for the food reward robot was written using MATLAB and C++. It was already in use by the time we began work on the project. However, the code has been worked on during our project by a third party, Aaron Winkle, due to some issues that were there before our team began work. This code has been used as a basis for future coding endeavors in this project, as we have been using MATLAB and Arduino coding in order to make our new additions interface easier with what has already been completed.
The functionality of the robot has been accomplished thanks to the work of the personnel working on this project prior to our involvement. Our team was tasked with the optimization of the robot by decreasing the total weight, removing unnecessary components, and increasing the traction by replacing the current tires.
It is important to mention that this is the first engineering design capstone at the University of Arizona that works in tandem with neuroscience research. This project has the potential to have a global impact in neuroscience, demonstrating the importance of human-centered engineering practices to aid other scientific fields in solving the most pressing matters impacting humanity. Alzheimer's is a disease that affects over 55 million people worldwide. It is vital to find better treatment methods and ultimately a cure. Through our contributions to the Evelyn F. McKnight Brain Institute, we are moving towards a world without Alzheimer's. And understanding the normal aging brain is the first step. Once again, we are Team 23089. We look forward to seeing you on Design Day.